Welcome, everyone. Frank LaFerla, Dean of the School of Biological Sciences. And before we get started with our panel, let me give a special shout out to the wonderful folks in the BioSci Dean's office for helping put together this great event. In particular to Shannon, Victoria, Rasan, Matt, Eric, Wes, Regina, and Lizzie. Just want to send my gratitude to you all. We have a few quick housekeeping notes. So the chat function is disabled for attendees. You can enter your questions through the Q&A function. This program also offers closed captioning. If you prefer, you could turn off the closed captioning by clicking the CC button at the bottom of your screen. And just so you all know, this program is being recorded, and the recording will be posted on our BioSci website for future viewing. It's been a little bit more than a year since many folks first learned about coronaviruses. I learned about them as a PhD student in microbiology at the University of Minnesota. And as I recall conversations with my colleagues about how fascinating these viruses were from a virology standpoint, it was often followed by the statement, quote unquote, that they just don't seem to cause any interesting human diseases. That was back in the late 1980s, and obviously things have changed markedly since then. We know that viruses cause profound human misery and death and catastrophe for our society. It is unimaginable that a year ago we would find ourselves in a position that over 450,000 people in the United States and over 2.2 million people worldwide would be deceased as a result of this virus. Whether you've been infected or not, we've all seen firsthand that SARS-CoV-2, the virus that causes COVID, affects us either directly or indirectly, and sometimes both. As I reflect back on my own adult life, it seems like viruses like HIV, hepatitis, West Nile virus, Ebola, Zika, chikungunya, SARS, MERS, and other infectious agents like mad cow disease have had a really devastating impact on our society. As we now enter into the 7th year of these Dean's Distinguished Lectures, one of our major goals has always been to unpackage complex topics in the life sciences for our community. And I am really pleased to see not only that we have a large audience from Southern California, but from across America. And we are here to address all of the major questions that are on your minds about COVID, including the new variants that are emerging, the efficacy of the vaccinations, and whether or not it's going to be possible to generate long-lasting immunity. But now I'd like to introduce our distinguished panelists. We should all give them our respect and admiration for all of the work that they have done on the front lines here, sometimes at great personal risk to themselves. So Professor Michael Buckmeyer is a faculty member in both the School of Biological Sciences and the School of Medicine. He is a former associate director of the UCI Center for Virus Research and former deputy director of the Pacific Southwest Center for Biodefense. And Mike has been called upon as an expert since the start of the pandemic, as he has a long history of working on coronaviruses. Professor Ilhem Messaoudi is a rising star in the School of Biological Sciences and has also done many projects related to COVID. She's the newly appointed director of the UCI Center for Virus Research and recently has been featured in several news and several local and national media interviews, including today in the Associated Press. So Professor Tom Lane was our most recent distinguished lecture speaker this past November, talking about the neurological impact of COVID-19. He's a highly influential researcher and has worked on coronaviruses throughout his illustrious career. Professor Andrew Noymer is an associate professor in the School of Public Health, or soon to be School of Public Health, in the Department of Population Health and Disease Prevention. I have to admit, I didn't know Andrew before this pandemic started, but now I'm really glad I do. He's been a source of incredible information. And so, not surprising, he's also been frequently quoted and interviewed in both regional and national media outlets. And last but not least is one of our most highly regarded alumni, John Gerace. He is a tireless advocate for the School of Biological Sciences and is coincidentally at the epicenter of global COVID testing, since he's the president of DiaSorin Molecular, which manufactures and distributes innovative diagnostic products. So welcome to all of you. We're all giving you a collective round of applause that you can't hear, but why don't we start with you, Professor Buckmeyer?
I should be on now. There we go. Well, it's a great pleasure to be here today. It's one of these things that that I never would have dreamed a year ago that that we would be caught up in a global pandemic of a virus that has been pretty passive so far. But this one is not. This is a real world pathogen. Our work has focused on these coronaviruses since the early 80s, and we've studied the function of the viruses, and we've studied a number of functions. But there's just 2 points that I want to make today, and let me go ahead and share screen right now. And we have, the first point is one that probably you've heard about, and if I can minimize this a little bit. There we go. You've probably heard more than you care to hear about the spike, but this is an image that my postdoctoral fellow Ben Neumer and I consulted on with this company Visual Science. And it's considered the highest resolution structure that we have of the coronavirus. It's because of this image here, it's kind of fondly referred to as a Death Star. So think of it as a very lethal virus, which has its own inherent beauty, but at the same time, it has great risk. These features right here are the star of the show, and they're in red here, and they're decorated with these small protrusions, which are glycosylation, and they're inserted into a membrane, which consists of protein, phospholipid, 2 proteins, phospholipid, and then inside that, I think you can probably just barely see it down here, we have a nucleocapsid. So in reality, we're dealing with a rather simple entity. It's not simple necessarily for viruses. It's in fact a very complex RNA virus, but it's a virus that gives us multiple challenges. And the first challenge really is how to attack this Death Star, because as we've seen already, you know, this spike protein, which is the key element in selecting the receptor and entering the cell, is capable of mutation. And those mutations may be mutations that make the virus grow faster because it grows higher in the upper respiratory tract, or because it's able to enter the cell more efficiently. There's a whole variety of different possibilities there. But the other thing is that those mutations also can be irrelevant. They may be just neutral mutations. And so we have to learn how to segregate those 2 problems. And I'm glad that, that John is here from DiaSorin, because one of the things that we don't do enough of is we don't do enough sequencing in this country. The last statistics I saw have placed the United States in 42nd place worldwide in terms of statistical analysis of these viruses. Number 1 was England. And so you have a great opportunity there to begin to understand what's going on a moment by moment basis, rather than looking at last year's news. And so that's the challenge number 1. Challenge number 2 is something that I think is kind of unexpected in terms of people who've worked on antiviral drugs. But this virus is capable of killing the cell almost immediately. And so if you're thinking about giving a drug that will inhibit translation, or inhibit transcription, or inhibit viral RNA copying, you almost have no chance to do that. And I'll show you the reason why. This is a basic, very simplified version of the entry and spread pathway in the cell. And basically what you do is the first protein that is made is called NSP1. And NSP1 is a protein that shuts down host translation. So that the host will no longer make proteins once NSP1 has been synthesized. You have then a whole series of other proteins that are made. And one of the things that happens is that you produce these double-walled vesicles. And these are the double-walled vesicles. And the purpose of these double-walled vesicles is to isolate the viral components from any cell components, including the innate immune response. And so you have these double-walled vesicles, and inside these vesicles, you have the production of all of the components that are necessary to produce virus. And it becomes basically an assembly line. And that assembly line will end up feeding virus into the urgic here, and then finally into smooth-walled vesicles and externally. And so you have basically the whole viral production is isolated from the cell. The cell is put out of action right away. And if you have those two things acting for you, you've got a substantial advantage. And that's exactly what happens. That's why this virus can be so destructive. The other thing that the virus can do is that it can change its mode of spread and entry depending on pH and depending on the cell types it's involving. And one of the things that we did years ago was show that by passaging in olfactory bulb cells, we could select for a virus that no longer entered the cell at the neutral pH fusion, which is the event that you see here, but instead entered by a deep endosomal entry. And that endosomal entry required acidification. So all of a sudden you change the character of the virus at the fundamental level. And so these are things that we haven't seen yet in COVID, but they may well come up. So I'd just like to stop there and answer questions.
Okay, Mike, let me start off with just two quick questions for you, and then we'll come back after all the other panelists have had a chance to present. But I guess, the first question is, how likely are we to, be living with SARS-CoV-2 in the population after the pandemic subsides? I think all of us are hoping that it just magically goes away, but what's your take on this?
Well, the magic was supposed to happen last June, remember? When the hot weather came.
That's right.
It's not going to disappear. It's going to take human effort to make it disappear. And it's going to, it's very different from SARS-1 in the sense that SARS-1 doesn't spread as avidly, but SARS-1 was much more deadly. Mm-hmm. SARS-1 killed 10% of the people that infected. And so that was an epidemic that really got the attention of the world. And we didn't yet, 18 years ago when SARS-1 hit, we didn't yet have social media. Mm-hmm. And so we didn't have all this confusion, all of this conspiracy stuff that's going around, and all the confusing things that people have weighed in and decided they are not going to be vaccinated, or masks are bad, or all of these other myths that you hear. What I tell my students is basically that a mask is a seatbelt. Mm-hmm. When you go outside or you go into a building and you put on a mask, you're doing the same thing as putting a seatbelt on in the car. It's no different. It's no reflection on your personality, no reflection on your manhood. It's just a precaution that can save your life.
Absolutely. Well said. So I've heard it said that we're likely to go from this being a pandemic to endemic, something that we're always going to have to live with that. Do you agree with that?
I think that there's some truth in that, because there will always be pockets of the infection, even if we vaccinate, and we're not going to vaccinate 100% of the US. I mean, mm-hmm. We're going to be lucky to get 50% the way things are going right now. But if you look at places in Brazil, like the city on the Amazon, a city of 2 million people has been hit 3 times, even though they have virtually 100% positivity rate in terms of antibody. They've been hit 3 times, three separate infection boosts. Andrew can put that better than I can, but 3 separate times, and you would expect it to flush through and then go on. Mm-hmm. Even smallpox did that, right. But it is not doing that, and it's a respiratory infection, which is the most easily spread kind that we deal with.
Okay. Well, thank you, Mike, for that. We'll come back because I see there's a lot of activity going on in the chat. So there are a lot of people have questions about the evolution of the virus. So we'll come back to you. In the meantime, Professor Ilhem Messaoudi is our next presenter. So Ilhem.
Hi, Frank, and welcome everyone. And Dr. Buckmeyer did a great job of presenting you with the virus. And my job is to present you with the yang of that yin and talk about our immune system. So thankfully, we have an amazing array of cells and molecules that our body deploys at the earliest sign of an infection to help us fight these pathogens, whether they are SARS-CoV-2 or just the seasonal influenza virus. And it all begins with early events. As soon as the virus enters the cell, any cell, there are sensors all over these cells that are just ready to detect that viral RNA or those viral proteins and leap into action. Any infected cell will make these molecules that we call cytokines. They're basically little breadcrumbs and beacons to help bring in immune cells, such as these monocytes and macrophages and dendritic cells and neutrophils, which I'm sure all of you have become more familiar with them than you really wanted to. Well, these cells come in here and they start basically attacking all these infected cells. And then eventually they bring in the big guns, which are your T cells and your B cells. And the T cells can further eliminate those infected cells, and our B cells make antibodies, which I think also everyone now is familiar with. So the virus is clever, but our immune system is also clever. And the concept of vaccination really leverages the fact that we have this amazing array, an organ system called the immune system, and leverages and harnesses that energy in order to protect us from an infection that we haven't encountered yet. So obviously this unprecedented pandemic has unleashed a tremendous amount of efforts to make several vaccines. There are upwards of 70 vaccines that are in various phase 1, phase 2, phase 3 clinical trials. They range in multiple variants from inactivated virus all the way to protein-based viruses. These efforts are primarily funded by private and industrial, but also in partnership with academic and public and nonprofit centers. Probably what everyone is more aware of and knows more about is these nucleic acid vaccine base. So this is Pfizer and Moderna. As you know, both of these vaccine platforms have received emergency use authorization recently. I actually received my second dose of Moderna almost 2 weeks ago. These are RNA molecules that are encapsulated in a lipid coat so they can enter the cell and also protects the RNA from being degraded. It comes inside the cell. This RNA is then turned into protein. The protein that your cells make is used to activate the immune system, and your immune system makes a response that protects you from future exposure. These two vaccines have shown amazing efficacy of 96%. For Pfizer, it's regardless of age. For Moderna, it's a little bit of drop for those over the age of 65, but it's really still amazing statistics compared to influenza seasonal vaccination, for instance. We also have viral vector vaccines that have gone pretty far in the trial. In this case, we take another virus, we modify it to express the protein from SARS-CoV-2 that we want to immunize against. Because this is another virus that has the ability to enter our cells and replicate in them. And again, that viral protein of interest is picked up by the immune system, and we generate an immune response that protects us from infection. Probably heard through the news that last week Merck pulled out of the market. Merck was making a recombinant VSV vaccine that expresses the full length of spike. It pulled out because it didn't get good clinical responses. However, there's a reason for that, and I think they'll come back. And then the Johnson & Johnson vaccine is based on an adenovirus. It's a recombinant virus, but it's not replication competent. So it means it cannot replicate, it can just enter cells and then help your body make that spike protein for the immune system to respond to. And Oxford AstraZeneca vaccine. Both of these are a little bit lower in terms of efficacy compared to the RNA vaccines. They're in the 70%. There's some interesting findings with the AstraZeneca that we can discuss if we have time during the Q&A. And then protein-based vaccines, this is exciting. So in this case, the protein is made outside commercially and then introduced via injection into the body. It's picked up again by the immune system, processed and presented, and an immune response is generated. The leading candidate in this category is Novavax, made a full-length prefusion spike protein. And the initial data shows that it's 85% efficacious against even the UK variant and 50% against the South Africa variant. So this is this vaccine platform, this one and this one are probably very close in line of getting the emergency use authorization as well. So that's it. That's all I have. I'll stop right there.
Okay, Ilhem, I guess you get the question of the day that's on everyone's mind. You sort of alluded to it, but that is, are these vaccines going to work against the emerging variants?
Yeah, so my answer is they will work to some extent. And even if they're not 100% match, I want to remind everyone that your immune system is amazing and capable of doing amazing things. And so even if it's not 100% match, the vaccine will train your immune system, will partially protect you while your immune system kicks into high gear and protects you from the new variant. So regardless of what new variants are in circulation, these vaccines will still have a significant impact on our healthcare.
Are we going to have to get annual booster shots, do you think?
Yeah, so that's a really great question. So far, we only have data for 5 months out, and it looks really good. So at 5 months, it doesn't look like it's decaying very much, but the jury is out indeed. So we're watching very carefully.
Okay, and we have a question in the chat. Someone wants to know, what's the maximum allowable time between the first and second dose of the vaccine to give the best protection?
So depending on what vaccine platform you're going with, for the most part, it's somewhere between 21, 28 days is what the clinical trials were based on. But there's actually interim data that shows you can go 42 days between the 2 shots and still get very good efficacy.
Great.
So we will have a little bit leeway.
So there's a little bit of wiggle room there. Thank you. All right, we'll come back to you, Ilhem. Thank you very much. So we're going to now move on to Professor Tom Lane.
All right. Thank you very much, Frank. Good afternoon, everyone. Thanks so much for attending this public forum. I think these things are extremely important. So let's see, I'm going to go back here. So our lab is very interested in studying the neurologic disease that's often associated with COVID-19. And as Frank mentioned at the beginning, our lab for over 20 years has been studying mouse coronaviruses and their ability to infect and replicate within the central nervous system. And we've been particularly interested in evaluating or characterizing immune responses that help control the viral replication within the brain, but also the ability of these mouse coronaviruses to persist in the central nervous system and cause chronic disease. And so last year, when COVID-19 erupted, we were able to take advantage of our experience in working in these models. And we've now got a very robust research platform in our laboratory to study some of the questions that we're very interested in with the ability of SARS-CoV-2 to get into the central nervous system and potentially cause disease. And why this is very interesting is because with SARS-1, which occurred in 2002 to 2003, and then MERS-CoV in 2012, there was some CNS involvement and some minor neurologic complications, but nothing like what we're seeing now with SARS-CoV-2. And so some of the questions we're addressing, I'm just showing some data in my lab that we just actually got this week, is can the virus infect and replicate in human cells of the central nervous system? And so these images here are showing at high magnification human neurons that were given to us in a collaboration with Dr. Leslie Thompson. And the neurons are shown here in red. And then we infected these neurons with SARS-CoV-2. And then we did staining to detect where the virus is. And so as early as 24 hours post-infection, we can detect viral protein in the cell bodies. And here's 2 neurons here that are illuminated. And then if you go out just to 48 hours post-infection, you can appreciate there's a vast increase in the number of cells that are infected. And what's particularly interesting to me is you're seeing the virus is extending down these long processes that are extending out of the cell body of the neuron. And so right now, these preliminary data are supportive of other studies that would argue that neurons certainly can be infected by the virus. And so we're extending these studies now to look at other resident cell types of the central nervous system. So these are the experiments we're doing with human cells. But then we also have a very active program looking at SARS-CoV-2 infection of susceptible mice. And so what we do is an intranasal inoculation of mice with SARS-CoV-2, and then we can collect brains and spinal cords from these animals at defined times post-infection and look at the spread of virus within the brain, and then identify cell types that are infected, and then some of the neuropathology or disease of the brain that might ensue. And so this is a low power image of a mouse brain. It's a little bit cut here, I apologize for that. But in red here is where we're detecting the virus, and it has very distinct anatomic locations. So in the cerebral cortex, and then these other areas of the brain right here. And if you look under high power magnification, what we see is somewhat of a replication of what we did, or what we determined with the human cells, is that neurons, at least in the mouse brain, seem to be a primary cellular reservoir for the virus. And so right now, we're characterizing the immune responses that are occurring in the brains of these animals, and more importantly, some of the neuropathology, or how does the virus infection impact the brain over a long period of time. And I think this has particular relevance because of this population of people that's growing called long haulers. And these are people that have cleared the virus, but then they have an array of symptoms that persist up to months. And many of these symptoms are associated with neurologic complications. And so I think this is a very important area of research, and this is another key area that we're trying to investigate. All right, so Frank, I'll turn it back to you.
Okay, Tom. So I guess a couple of questions that on everyone's mind is, what if you have comorbid conditions, particularly of the CNS, like Alzheimer's disease or multiple sclerosis? Is that going to make your COVID worse?
Frank, that's a great question. And so that's something we're doing right now. As you know, we're looking in several different avenues. We've been able to collect brains from patients that have Alzheimer's that passed away from COVID. We're looking to see if there's an increased CNS penetrance of the virus, as well if there's either an increase or a decrease in the neuropathology associated with that. And that's certainly true also with MS. And we can model these things. We actually have experiments going on as we speak, where we are infecting transgenic animals that have an AD-like pathology with our mouse coronavirus to see if there's an impact on disease. We're also doing the same with in models of multiple sclerosis. As you know, that's another big area of work in my lab. So, you know, I think this can extend to a number of different chronic neurodegenerative diseases outside AD and MS. So we're very interested in pursuing that.
And I know it's still early days, but when we say long haulers, exactly how long are we talking about?
Well, you know, there's been a flurry of papers that have come out. The NIH has really brought out a beautiful platform looking at this. You know, it's not clinically defined yet, but we're getting more parameters on this. And so how they define this now is if you have an array of symptoms up to, I think it's 28 weeks or something like that after infection, that's how you're defining it. And so we don't know how long it'll go, right? As you pointed out, the disease is still pretty young. But if you think about the number of people that have been infected and survived either severe or benign disease, and they have these long hauler symptoms, this could be a real problem. And we need to really start interrogating what the mechanisms are. And so the animal models are great, but it's really challenging to, like a mouse, to study some of these things, but we're trying.
Good. Well, thank you for that, Tom. So let's move on to Professor Noymer and his presentation.
Thank you. Thank you for inviting me, Dean LaFerla, and thank you to your staff for organizing this, and thank you to everyone watching this at home. It's a great pleasure to tell you a little bit about my work on COVID, and I have no conflicts of interest to report. So just over a year ago, I tweeted something, and I tweet every day about my current thoughts about the coronavirus. I'm at Andrew Noymer on that platform, and I tweeted that COVID was going to be like a really severe thing that was going to, impact society. I used some interesting language to describe that sentiment, but basically I said, this was going to be a big deal. And why did I feel, in January of 2020, that COVID was going to have a significant impact? And the reason is I study pandemics as part of my work in public health, and in particular, the 1918 influenza pandemic, which was the subject of my PhD dissertation. And I knew in one respect that this was actually going to be worse than the 1918 flu, and that is that we are immunonaive to this coronavirus. So, there's been a lot of interesting work sort of talking about the ways in which there may be cross-reactivity with other coronaviruses that we do have experience with. But by and large, experience has shown in the last 12 months that most of the world's population is significantly immunonaive to this new emerging coronavirus. And in 1918, there was an emerging strain of influenza. It was H1N1, and that caused a lot of morbidity and mortality. But there was influenza before 1918. So influenza, generally speaking, wasn't a virus that the world was immunonaive to. It was just the H1N1 strain was new. And this effect caused a peculiar aspect of the 1918 influenza virus, which was very deadly, but it was deadly in unusual age groups for influenza. It was deadly among 25 to 45-year-old people, whereas influenza normally is most deadly among infants and the very old. And so it was H1N1 was an emerging strain in 1918, but it had this peculiar mortality pattern, which was influenced by cross-reactivity to previous influenza strains. So elderly people in the 1918 influenza pandemic actually didn't die at rates that were any different than a normal flu year. And the reason for that is that there were strains of influenza circulating in the 19th century to which these people had already been exposed, which conferred them some degree of protection compared to younger people. So that's something that's different here. The whole world is, for all intents and purposes, or at least 12 months ago, for all intents and purposes, was immunonaive. And that means that the virus has a wide field to run. And that means that I knew then, and as I still feel now, that this is going to be a major event. And as we've seen, it is a major event. And so it's important to think about that. And another thing is that you may have heard that the coronavirus isn't much more deadly than influenza. In fact, it is about 2.5 times more deadly than influenza for all groups 18 and up. And it has a very curious low mortality for children, which we don't really fully understand. But, it's in the same ballpark as influenza, although it is more deadly, but it will affect more people than a given flu year because of this immunonaive aspect. And in fact, it will affect more people in 12 or 24 months than 5 or 6 flu seasons worth of influenza exposure because of this immunonaive aspect. And so even though the mortality, as Mike said, is less than SARS-1, it's going to be a significant event. We need to think in terms of the fact that 12 months ago, 99% of the world's population was immunonaive. And this came up earlier, but we're not going to eradicate COVID-19. It's going to change. We're going to be having COVID-19 a year from now, 5 years from now, but it won't be the same as now. The H1N1 flu that emerged in 1918 was the dominant influenza A strain until 1957, when there was another flu pandemic, which introduced a new strain of influenza H2N2. And so, we're not going to eradicate COVID. I mean, it's just not going to happen. The genie's out of the bottle, so to speak. So, our vaccine is still our best hope to control it. And I really enjoyed the remarks about the vaccines, and the vaccines will absolutely help us, get the upper hand on the virus, but it's not going to be eradicated entirely. And so, we're just going to it's going to be something that's going to have staying power. And I think my 5 minutes is up. So.
Okay, and Andrew, you alluded to this about the lower mortality rate or fatality rate in children. So the question that I know a lot of parents have is, when is it safe to send their children to school? Does everyone have to be vaccinated before they can do that?
That's a very important public policy question right now. So the vaccine has been trialed in people 18 and up, and it has been authorized for use in has been authorized for use in 16 and up. And there are studies underway now, pediatric trials, that will eventually lead, we believe, to authorization of the vaccine for children. But right now, the vaccine is not authorized to be used in kids. So I don't think that's the criteria before which we can send kids back to school. It would be great to see teachers and adults who surround kids to be vaccinated, and then we'll be able to get things going a lot more back to normal.
Okay, thank you. Let's move on to John Gerace. And, I should also indicate, John, your most important accolade is that you're a member of the Dean's Leadership Council. So thank you for your service and everything you do.
Thank you. Excellent. Well, listen, I am obviously the one non-academic person here running a business. So I want to apologize for that right off the bat, for profit. So that's my disclaimer. Just give you a little background, DiaSorin, as Dean LaFerla indicated, we're a global diagnostic testing manufacturer. We don't do the tests, we make the tests. We've been looking at and studying and developing tests for infectious diseases for over 40 years. We have about 3,000 employees worldwide, and I run just the molecular business unit or subsidiary of DiaSorin. And so us as a company, we have all different types of tests for the SARS-CoV-2 virus and COVID-19 disease. The molecular test that's based upon a polymerase chain reaction, PCR. It's probably the most accurate test you can get. It confirms you have active infection. And then our company also produces an antigen test that is, again, it confirms you have active infection. It's a little less accurate than PCR. It looks at a viral particle that comes from the spike protein. And then an antibody test, and that is confirms that you had the disease, right? So our company provides all three. We were one of the first companies to actually come out with and get emergency use authorization for the COVID-19 test we have. It was a little bit serendipitous. We were studying respiratory pathogens coming out of the new product. We have a flu product out on the market. We've have all different types of respiratory pathogen tests. And it happened to be that our scientists in Italy were looking at different pathogens and came across this Wuhan strain. And right within days of its viral sequence being uploaded to GenBank, our scientists had designed a PCR-based test for the detection of the SARS-CoV-2 virus. So that was a really, again, serendipitous moment to have A, looking for a product at that time, and B, being in Italy. About half of our R&D group is in Northern Italy. And you all know that was a leading indicator of what was to happen because of the acceleration of the spread of the disease happened in Italy before the US. So that is where we stand. We have a test right now that we've ramped up production to. We produce over 1 million tests a month. Our test is a really unique form factor. It's 8 samples get loaded onto what looks like a DVD. It gets put into a little device that's no bigger than a shoebox, and you get positive results within 25 minutes displayed, and confirmation of a confirmed negative within 90 minutes. So we've been, among a lot of companies now on market with testing. The last time I looked, there was almost 200 companies that had some form of molecular test that has been receiving emergency use authorization. So our claim to fame has been, we've really focused on supply chain, production, and the instruments that we've placed for COVID-19 testing, we've had 100% service level, meaning that if we said we're going to ship 10,000 tests a week, we ship 10,000 tests a week. And now we've doubled our installed base, and we still see in 2021, even with vaccines, the criticality of getting tested just to monitor the disease and making sure that we do get the herd immunity that we're all anticipating. So that's who DiaSorin is. That's what we do. We're here right in Orange County, and we very proudly employ a lot of really smart molecular biologists from UCI School of BioSci. So every time we hire a new UCI employee, I ring a bell. Okay, so zot, zot, zot.
Well, thanks for that, John. So, just tell us, what's your life been like recently? I mean, the challenges that you must be facing are just incredible, just scaling this up. So can you give us a sense of that?
Oh, yeah, it's 24/7, and you don't want to meet our VP of operations, Vincenzo Albano. He's worked magic. He scaled up our production like 800%. Just amazing what our ops team, and not just ops, we've had to handle all elements of our supply chain and how we serve our customers, service, order entry, quality. It impacts every aspect of the company. And what I'm most proud of is how we all have responded just valiantly to the task. It's been amazing.
So, I guess another critical question is the testing and the variants. Will the tests pick up the new variants?
Our tests will pick up whether or not you have the virus despite the variants. Okay, so most good tests on the market, they're designed by targeting different gene targets within the viral genome. We do 2 different gene targets, and that way, again, if there is a mutation, there is some level of redundancy of what you're targeting, and you'll pick up the virus. There are definitely tests that will tell you what variant you have. They're sequencing based.
Mm-hmm.
And as Dr. Buckmeyer mentioned, sequencing is extremely critical. There has been a new bill right now proposed to fund the CDC another $2 billion in sequencing surveillance. We've analyzed over 50,000 viral sequences to date just to keep on top of the variants, and our test is very robust against all of those variants.
Awesome. So let me invite back all the other panelists so we can open this up, because there are a lot of questions that are in the chat here. And so, I'll apologize in advance if we don't get to all of your questions. But I guess one of the first ones that I see here, and Ilhem, perhaps you're the best one to answer this, is if you're pregnant, should you consider getting the vaccine or not?
Yeah, I think that's a really great question and something that I'm very passionate about. So pregnant women are extremely vulnerable to complications from COVID-19. The data is very serious out there. They are more likely to end up in the ICU due to reduced lung capacity. You're immunocompromised to some extent because you have to carry this foreign object to term. I'm a mother of 2 kids, so I know something about this. But it's really, because of that, it's incredibly important that we encourage pregnant women to get vaccinated. I get this question a lot. If I'm nursing and pregnant, should I get vaccinated? And we encourage women to get Tdap vaccines. We encourage women to get the flu shot. And this does 2 things. It protects you, but we have this wonderful thing called passive IgG transfer through the placenta. And that means that not only are you protecting yourself, but you're providing that baby with a dose of antibodies that can protect him during the first few days of life, up to potentially 6 months of life. That's how long maternal antibodies can stick around. So while we don't have a lot of data, OBs around the country have been encouraging pregnant women to get vaccinated.
Good, that's very clear. So what about the thoughts about reinfection, where patients no longer show any trace of the virus with PCR, and then can they get reinfected and have a positive viral test? Maybe John, you might want to answer that, or Andrew. How likely is someone to get a second infection again?
Andrew, you're muted. Oh, there we go.
Well, I mean, the data we have on that is still kind of a question mark. I mean, the numbers of infections that we're seeing in places like Manaus, Brazil, seem to suggest that reinfection is happening because the numbers of people and waves that they've had, wave after wave of infections that have occurred in Manaus, are profound. Some of the early estimates of seropositivity in blood donors in Manaus may have been overestimated, which may, bring down the extent to which we're seeing multiple infections. But particularly with the emergence of the variants, we know the so-called South African variant evades some of the antibody therapies that we've had. So that's also evidence that it may be capable to create a second infection. So it's definitely something that we're monitoring closely.
Okay, Mike, perhaps you could answer this next question about, what are the molecular differences between SARS, MERS, and SARS-CoV-2?
SARS-1 and SARS-2 are very similar. The original estimates were something like 75 to 77% identity. I think that that's an overestimate now because the viruses have been drifting away. But keep in mind that there are a lot of mutational changes that occur that have no effect on the biology of the virus. The virus is continually making mistakes in replication.
Mhm.
In terms of MERS, MERS is quite a different virus, and it probably only bears about 30% similarity, but it does have the same genome organization. So it's presumably something that has evolved along a different track, and it uses a different receptor as well.
Mhm.
So you have a number of things that suggest that it's a different virus. One thing that's a little bit frightening about these is that, as we saw from Ralph Barrick last week, that there are some mutants which now are appearing to be recombinants between human, relatively low pathogenicity human viruses and animal viruses.
Mhm.
And these are starting to show up as well. And one of the favorite targets of that is OC43 combined with the pig virus. So if that starts happening, that's a whole different stream again of evolution and entry into potentially into a human life cycle. So just keep a watch on that.
Okay, Tom, here's a question about the nervous system, and folks are wondering, can the immune system really get access to the CNS tissue to attack the, viral infected cells or free virus?
Yes. So in our mouse studies with the murine coronavirus, it's very clear that activated immune cells, both from the innate and the adaptive immune response, can rapidly accumulate within the CNS and control viral replication through a variety of different mechanisms. If you look at some of the histopathology that's been reported within the brains of COVID patients upon autopsy, you do see lymphocyte infiltrates in some of these patients. So lymphocytes are components of the immune system. You also see microglia activation. Microglia are the immune cells of the CNS, but also other types of immune cells in the periphery are able to accumulate in the brains. But there's no clear neuropathology. It's kind of across the board. So I think that's probably dependent upon a number of things, potentially even the initial viral load, comorbidities that might be existing that we all know about if you have cardiovascular problems and metabolic disease. So these are questions that we're still that are very important, but we're trying to address. But the immune system is very good, as Ilhem pointed out, at working on your behalf and trying to get rid of this virus.
Good. So here's a question about the UK variant, and someone wants to know, why are children more just as susceptible as adults to getting it, and why is the UK variant more infectious than the original version of it? Maybe Mike or Andrew or Ilhem, one of you can handle it.
I actually heard a paper about this last week.
Oh, good.
At the Cold Spring Harbor meeting, and what is theorized at this point is that what's happening with this UK variant is that it's a variant that does not appear to make the disease more deadly, but what it does is it replicates more in the upper respiratory tract and the nasopharynx than it does down deep in the deep lungs. And so what you have is this pattern similar to what you see in a child.
Mm-hmm.
The child tends to do the same thing, at least from the orders that we've seen. And so what may be happening is that the virus is selectively growing higher in the respiratory tract, makes it easier to spread because it's easily coughed out or whatever, spurtled out, whatever. And at the same time, it can infect both adults and kids.
Mm-hmm.
I just want to add one thing. So to clarify, so kids get infected. So we want to dispel any myths that kids do not get infected with this
No,
virus. They just do not have the same clinical presentation as adults. So kids are just as susceptible to infection. They somehow are refractory to severe disease. And that's because of some of the immaturity that comes with their immune system that's actually working in their favor in this case.
Mhm.
Here's another fascinating question from the audience, and that is, how many people, or the percentage of the population needs to get vaccinated for us to bring this under control in the United States?
Well, I mean, probably around 80% of the population needs to have some sort of immunity. I mean, and that can come from vaccination or from surviving natural infection. Now, we already talked about reinfection, but typically surviving natural infection will confer immunity. So, but 80% would be the herd immunity threshold. But it could be a little bit lower. It could be a little bit higher. A lot will depend on this moving target and how many of these variants come down the pike and I think nobody knows for sure the answer to that question yet. In the long run, I'm quite confident, long run, speaking of years and decades, that that new variants will be our friend, that will have a lower equilibrium between the human population and its immune status and the virus. But in the very short run, as we've seen, some of these variants can cause us headaches. But I mean, figure 80% would be my guess.
And what's your best estimate as to how long it's going to take us to get to that 80% threshold?
I mean, that's going to depend on a number of things, both logistical and as Mike alluded to, people's resistance to being vaccinated, that may or may not be, encouraged by social media. So I would hope that by the end of the summer of 2021, every adult who wants a vaccine will have been offered a vaccine. But
so, John, can you talk to us about testing in third world countries and what's that like?
Well, there's, with all sorts of infectious disease, there's a challenge to get really low cost, portable, tests to developing countries. And although our company doesn't focus in that area, I could just speak to how much, the Gates Foundation and other organizations, even the US government, the Bruce Thomberg at NIH, the RADx program, very impressed with all the money and the efforts in developing really, really low cost tests that are portable for developing countries.
So, Ilhem, here's an interesting vaccination question. And someone writes, if this is a respiratory disease, why don't we immunize through the nose to elicit secretory IgAs by local cells to stop the infection early on?
Yeah, and that's a really, really good question. So there's actually been some attempts at doing intranasal vaccinations that have resulted in the opposite phenotype of what we were hoping for. So some of the studies that have been published have shown that intranasal vaccination is less protective than an intramuscular injection, and then sometimes actually can exacerbate disease in animal models, of course. And that's why these studies did not go into any further than that. So they stopped at the preclinical stage. And that's something that we'll need to investigate a little more. I mean, part of the disease pathogenesis is the cytokine storm. And so, so an overexuberant, misdirected immune response, basically, that's what a cytokine storm is. And so there is some thought that an intranasal vaccination is then eliciting an aberrant, the second exposure will elicit an aberrant immune response in the respiratory tract, and that's not what you want, as opposed to IgGs that are circulating. And even it's a myth that things IgG cannot get into mucosal sites. IgGs, immunoglobulin G, can actually get into mucosal sites and do a great job of helping clear out viruses and getting the T cells to work as well. So.
Okay, so let's follow up on with another vaccination question. And I know it's early days and these data just are not, there yet, but maybe you could just speculate for us, or your best guess, what you think long-term safety concerns are going to be for COVID vaccines. And just piggybacking off that, someone else also has written whether or not, if you already had COVID, whether or not your response to the vaccine is more severe.
Okay, so I'll take the second one first, just because there's actually a really nice paper from Mount Sinai by Florian Kramer group that showed that people who have had COVID tend to have a few or more. So I had my Moderna shot and my arm really hurt, and Dr. Lane is actually feeling the effects of his booster shot today, which is why he's a little lethargic. So these responses are a little bit bigger in individuals who have had COVID, because they're actually essentially getting like a third booster and a fourth booster in some cases. So the recommendation is out that people should wait 90 days from the time of their diagnosis to actually get their vaccine. And part of that is not that there's any counterindication, it's actually dose sparing. It's helping save vaccines for those who really need them and have no immunity. So we know for sure that antibodies following natural infection are really high, persist at a high level up to 90 days after you've had your COVID diagnosis. And so we feel that you're pretty protected for that time. And so we're going to prioritize people who don't have antibodies. So it's a form of us for dose sparing. So there's potentially, like, you're going to get like these further amplification of your memory response. So you might feel that little bit like that, that flu shot feeling the next day. That might be a little bit worse for you. Sorry, Frank, I forgot about what the first question was.
That's okay. I think you handled it nicely. Tom, let's talk about long haulers again. Someone writes an interesting question here. Really unpackage for us what that really means. Does that mean that the brain and neurons are serving as a reservoir for the virus, kind of like a Trojan horse, or what's really happening when we say long haulers? You're muted, Tom.
Frank, it's a question I've been asking myself every day, right? And so, there's certainly evidence that you can detect virus in the brains, primarily in neurons, right? In cerebral cortex and in the olfactory bulb, right? But is that persistence, if you will, creating some of these symptoms? I mean, long hauler symptoms are a myriad of symptoms, right? They affect multiple organs. And, a lot of these symptoms are chronic fatigue combined with anxiety and depression, and then brain fog. One of our personal friends who got over COVID, who did not have it very bad, has had a real problem with this, right? With brain fog, to the point where she went to work and then came back to her car after her work, and she's like, why is my car still running? She forgot to turn off her car, right? So, I mean, you're seeing this in real time. And so my personal opinion is that it's probably combinatorial, right? Certainly we know that these coronaviruses can persist within the CNS, and that's never a good thing. But you can have these long-term effects, particularly in the lungs, right? That can secrete a lot of different cytokines and what have you. And these cytokines can be elaborated and get into the central nervous system. And I think, and to some extent, that's probably why we're seeing microglia activation, right? These cytokines get into the CNS and they can affect a lot of different things. And I'll conclude with this. There's a beautiful paper by Tony Weiss-Corey at Stanford that came out, I think, 2 months ago. And it was very elegant and a very precise study where he was looking at neurons from the brains of patients that had died of COVID. They weren't infected. He showed that the microvasculature was, but these neurons in discrete anatomic areas of the brain that control depression, areas that are associated with mental illness, were particularly affected. And so I think this raises some really fascinating questions. If somehow infection turns these genes on and off, and how long will these be on, right? So again, these are questions that are really challenging to model in animals, right? But we're trying to, move forward to address some of these. But it's a real sticky question, right? And if 10% of people develop long hauler symptoms, what's 10% of 100 million?
Yeah.
That's a real problem.
A big number. Okay, so this person clearly is tired of staying home and wants to know, how safe is it to travel internationally after getting the vaccine? So Andrew, you want to take that?
Yeah, sure. I mean, until it's safer than before the vaccine, and but until we have more of a better handle on it, countries are going to still have restrictions on incoming international travelers, and which in some cases will involve showing a negative test. And I haven't heard yet about to what extent of a completed vaccine certificate it will waive any of those requirements. So anyone who wants to travel internationally would do well to study up on that well in advance of the trip, and also to keep checking on the daily, almost even before they depart, because these restrictions can change very, very quickly, and you may spend the first 2 weeks of your holiday in a quarantine hotel, depending on where you arrive. And the other thing is that the airlines are unlikely to waive mask requirements for vaccinated people, at least during the phase in which most of the population is still unvaccinated. So be prepared to continue to wear PPE, and that would be my advice. But the vaccine is going to be the route back to the world we had before.
So someone asked about vaccine prioritization and wants to know how that priority list was determined. Was it to stem the high mortality rate among the elderly in the initial wave? And why haven't those with underlying comorbidities been prioritized as highly as well, regardless of age? So I think that's a very good question. Ilhem, do you want to take that?
Yeah, I mean, absolutely. So this is a question that a lot of us have asked. And so the reason for the prioritization is that our healthcare system has been under tremendous pressure. So my husband is a pulm critical care physician who has not had many days off recently and is covering for the community. So I can speak from a very personal space for this. Our ICU nurses, our ED doctors, our pulm critical care doctors, they can't take this anymore. They cannot run at 150% capacity anymore. They've been doing it for almost 10 months straight. No breaks, no holidays, double shifts, helping community. And so the vaccination strategy has been to stem the flow of people who end up in the ICU requiring a ton of supportive care. And those overwhelmingly tend to be people over the age of 65. People over the age of 65 account for 80% of deaths in this country. And so we're starting with them. And I think 60% of deaths are from individuals who are in nursing homes. So that's why we're starting with them. And we're starting with them, and we're starting with people who come into contact with older individuals. So the prioritizations, for example, for nursing homes and all the staff who work in nursing homes, because obviously the people who are in nursing homes are not the ones bringing the virus in. It's everyone who's coming in and out of that nursing home. And so we needed to have these ring vaccinations to really protect the people who have the highest chance of ending up in the ICU. And one other thing to keep in mind is the average stay of a person in the ICU pre-pandemic was somewhere around 5 days. The average stay of somebody with COVID in the ICU is 15 days, and in some cases 6 weeks. And so the ICUs are they require very intensive care. And if people are staying there for 6 weeks, that's a bed that's not available for a cancer patient, for somebody who has just had a car crash, et cetera, et cetera, et cetera. And so that was why the priority list was set the way that it was. People with comorbidities are actually not that far down the list. So we're very attentive to that. The ACIP, the Committee on Immunization Practices by the CDC, very much took that into consideration.
Okay, John, a question here arises about testing children. Since they don't get a severe disease, what's your take on the need to test children for COVID?
I think it's very important, and it's particularly children that are of school-bearing age, right? To keep kids in school, keep them protected. And I think that as we're looking at different sample collection modalities that make it easier for kids to get tested, saliva, nasal swabs as opposed to nasal pharyngeal swabs, and just making tests simpler, easier, less painful, less irritating, particularly for children.
Good. Mike, someone writes, will SARS-CoV-2 become less lethal as it evolves? And can we also use tools like artificial intelligence to try to predict which variants are going to emerge, and so we could get ahead of the variants by developing new vaccines?
If you look at the scientists, there's an article in the Scientist showing all of the mutations. I'm sure John's aware of this. It shows all the mutations, and then it shows the 5 or so that have made significant changes in the biology. So the question is, how do you associate the change with a change in biology that's worth reacting to? And I don't know what the answer to that is. And as far as artificial intelligence, we're having a hard time keeping up with the sequencing, and we need to generate data regardless of whether it's artificial or not. So I think that that's a long shot right now. It could develop, but it's a long shot right now.
So a follow-up question on that point, it relates to super spreaders, Mike. And can you talk about exactly what they are and what's different about these people?
They are, for example, one of the major justifications to begin immunizing children. Because children, who are asymptomatic, or adults who are asymptomatic, can spread the infection freely without even knowing that they're infected. And there have been many super spreader events that have begun that way. And so we have that example. I think that as you consider again what the changes are, SARS-1 did become less virulent as it circulated through humans. It became much less virulent. And so that was part of the disappearance of it, but it wasn't the sole cause. The sole cause was really the rigorous quarantine and social distancing and all the things that were done correctly. And that was done in an era when we had a smaller population, and it was mostly contained in Europe and Canada.
So have you ever heard of a virus being as promiscuous as SARS-CoV-2 is? I mean, it just seems that it can get into the CNS, into the upper respiratory tract, the lower respiratory tract, the GI. I mean, there aren't many viruses that are that promiscuous.
Influenza can do a lot of that. If you get into some of the substrains of influenza, you can get neurologic infections. We do see some viruses, but most viruses are very specific in terms of their receptor range, the acceptable receptors. And in fact, most coronaviruses are that way. And so a cat virus doesn't necessarily infect a dog. And that's the kind of politeness that we've accepted for years, with viruses. And this one's breaking all the rules because it'll infect your cat, it'll infect your daughter, it'll infect your grandmother. And so we have to learn how to deal with that.
Well, it's interesting you say that because someone wrote a question here. Can pets get COVID? And if so, should we vaccinate them? And when will a vaccine be available for pets?
Interestingly, cats get COVID, but they show no symptoms. They're little super spreaders running around the house.
Dogs do too, actually.
Yeah.
Okay, so we just spend a lot of time talking about the vaccines. What about antiviral drugs? What role do you think they have to play? And maybe Tom and Ilhem, you could take that question.
Ilhem, you want to go first?
Sure. Antivirals are absolutely critical. So especially in the face of not being able to, really rapidly vaccinate 50% of the population to get to 80% immunity for, we need antivirals, but they're going to be very tricky. So remdesivir, for all the press that it got, really is a drug that if it's not administered very early post-symptom onset is really not that effective. And then as Mike eloquently described, this virus has a really great way of building what we call viral factories inside cells that sort of shields it. So antivirals are just going to be really tricky in terms of being able to target stages of the viral life cycle that are easily accessible, where the virus is not shielded. But we should look into things like inhibitors of viral entry and other things. But they absolutely have an important seat at the table, and we should continue to develop them very aggressively.
Yeah, I can't really add to what Ilhem and Mike said, but one of the areas that we're actively pursuing in the lab are novel anti-inflammatories, right? So that you have maybe a slightly larger window of time. And so, we have experiments ongoing right now in the BSL-3 where we're treating infected animals with novel anti-inflammatory peptides. And I think those can be a very effective first line of defense in dampening down, particularly the severity of lung pathology, right? And then who knows, some of these drugs, if they're CNS penetrant, might be able to also dampen down any inflammatory responses in the brain.
And to Tom's point, dexamethasone has had a more significant impact on altering the course of disease than remdesivir has for about, a hundredth of the cost.
Wow, good. So John, someone is asking about the testing here and whether or not it can pick up defective virus particles, or does it have to pick up the full infectious particle on your test?
Okay, that's probably beyond me technically. I would assume it could be either, because we're only detecting portions of the viral genome. So I wouldn't assume it could be either dead or alive, right? These are short sequence, relatively short sequences throughout the viral genome. So that's the business answer to that scientific question. Sorry.
Okay, good. No, that's fine. So, we haven't spent any time talking about the origin of this virus. And Mike, maybe you want to talk on that, and Ilhem, you could chime in on this as well. I mean, did this emerge from bats? I mean, what's the latest thinking on that? In the community, we're constantly confused. We're here, it came from bats. It came from one of those markets, and now people seem to be moving away from that. What's the latest, greatest on that topic?
There's been a lot of attention paid to it recently. The investigation is reopened with WHO and the committees. The virus did originate with bats, but the question is, how did it get from bats to humans? It was being studied in the Wuhan labs of virology, and it also was found in the market.
Mm-hmm.
And so we don't know, we don't have a clear trace, unless Andrew has seen something in public health. We don't have a clear trace of how the virus got from the bat to the human. We do know that most of these viruses require an intermediate host to do that, but if this one is so capable, it may spread directly. But we don't know that. There's also a story that's circulating now about caves in Thailand where you can isolate the virus from bat guano. And if that's true, that's a whole new dimension. So we don't know yet, is the answer.
Okay. Ilhem, here's a different question. If someone already had COVID, should they get the vaccine when their name is called, or should they wait given the lack of availability for others?
Well, the CDC recommends that anybody who has had COVID wait at least 90 days before they get vaccinated. One of the questions that you will get when you get to your vaccination appointment is they'll ask you if you've had COVID and when you've had COVID. Mm-hmm. And I guess it just depends on how that vaccination site is run. But if they're following the CDC guidelines, they'll tell you to come back if you've had COVID in the last 90 days.
So Andrew, maybe can you comment on this question here? Someone writes, why are some people infected with the virus have virtually no complications, they're asymptomatic, and yet others have very severe complications?
Well, I mean, this is certainly a feature of COVID-19, but it's not that uncommon in respiratory viral infections. I mean, influenza can cause anything from a severe hospitalized pneumonia to a few sniffles and sort of anything in between. I mean, there are some viral diseases like smallpox, which, are 90% or more of cases cause symptomatic infection. But there are other viruses that it just cause a diversity of degree of severity, and it's not that unusual. It's just one of the perplexing things about COVID-19. Maybe Ilhem has an immunological perspective on that, but it's not unheard of.
Mm-hmm.
Yeah, I mean, every pathogen causes an array of symptoms that range from mild and remarkable disease to severe disease, right? So, I mean, yellow fever kills 30% of the individuals, but the other 70% are not dead, and they don't have, and some can develop natural immunity. I mean, we find antibodies to Ebola virus in people who never even had any signs of Ebola viral disease. So a lot of it is host genetics, your immune response, the circumstances under which you're infected, the amount of virus or pathogen you've been exposed to, and what you did afterwards, underlying health. So it's this very complex equation of factors that come into it and then that the outcome is the spectrum of disease manifestations. I mean, it's kind of interesting. Everybody thought at the beginning HIV patients, for instance, were going to be really, really hit hard by COVID-19. And they're very underrepresented, actually, if you look at the mortality data and the severe disease. And there's many explanations for this, right? They know they're immunocompromised. They take better care of themselves. They actually believe and respect public health because they know that science has been on their side. So there's that social behavioral aspect to it, but there's also the thought that maybe some of the antivirals are actually acting in some way to disrupt viral replication. It's an RNA virus. The other one's also an it's been surprising to us. And so some people on certain immunosuppressive drugs have actually seemed to have not go through that massive cytokine storm and actually have better outcomes than we initially thought they were. And on the other hand, I know people who are really young who have no comorbidities have been in excellent health and they ended up in the ICU. So there's really, I think we're just learning a lot more about the factors that regulate or modulate the disease severity, and we have lots to learn.
Yeah, and I would just build on that. There's some fascinating literature showing the people that make antibodies to interferon, right? And they have an increased susceptibility, or they have deficiencies in responding to type 1 interferons, which are key antiviral molecules that are expressed early in infection. But also to Ilhem's point, we have a panel of brains of patients that actually had AIDS that then passed away from COVID. And so discussing comorbidities, we're really interested in looking at, is there increased CNS penetrance to the virus in these patients? And is there increased neuropathology, right? And so there's a whole series of other questions that we need to ask and address. So again, it goes back that we're very young in this pandemic, believe it or not, although it seems like forever that we've been dealing with this. So.
And I'll add one more thing. We just had this very big study where we compared young and older COVID-19 patients who were in the ICU. So both had very severe disease, but when we compared the two age groups, the older individuals, their trajectory of disease was very different than that of young, which really explained why unfortunately they have more fatal outcomes and additional complications. But for example, things like the younger patients their T cell numbers were able to rebound faster while they were in the intensive care unit, whereas in older patients, those T cell numbers just kept on dropping the whole time they were admitted to the hospital. So there's all these interesting dynamics in terms of the immune system that are happening in the background that ultimately manifest themselves in these different outcomes.
John, another question regarding testing, and someone's wondering whether or not there are any tools or any tests that are in development to identify the long haulers.
We're right now just focusing on saliva and point of care. So I'm not familiar with. I think that I'm listening to the discussion, and as one of the things that we're really good at in other diseases, pathogens, is detecting pathogens in cerebral spinal fluid, and so anything that crosses the blood-brain barrier. So we'll keep an eye out on that. We have a pretty popular herpes simplex virus test that's off of really small volumes of CSF. So if that is a viable sample type, believe me, we'll be on top of that one pretty quick.
So another question, actually several individuals raised this, is the connection of this disease to blood types, both in terms of the infection and in terms of immunity. So Ilhem, you want to take that, or Andrew?
I really, that's a fascinating literature that I am not super familiar with. I've seen the associations, but I am not sure that there is, like real evidence-based discrepancies in disease outcomes. I don't know, Andrew, from an immunological standpoint.
Yeah, I was going to say almost exactly the same. I mean, we seem to see more severe outcomes in type A and less severe in type O blood in the ABO system, but it's not clear to me how robust that is. That's going to be something that we're going to have to keep monitoring as the pandemic unfolds. And if you test a lot of different hypotheses, a remarkable number of them wind up looking significant at first blush. And then as the evidence accumulates, you know, they really fall away. So it's not the overriding factor. Let's put it that way.
Yeah, usually those kind of hypotheses take a long time, and they tend to linger in the community for a long time, kind of like that Alzheimer's is caused by too much aluminum exposure, right? I mean, that's one of the questions that people always ask, even though there's not much data for that. So I want to be respectful of everyone's time, particularly since there are a number of folks on the East Coast here. And so why don't we go around and give everyone a minute or two to kind of, you know, just give your takeaway messages for the folks out there. And so we don't have to go in any particular order, but John, since you're in my top part of my screen, do you want to go first?
Sure, I'll just be an advocate for testing and wearing masks.
Okay, Andrew.
Sure. Well, thank you for joining us this evening, and thank you for supporting UC Irvine and the School of Biological Sciences. I mean, you know, the 1918 influenza pandemic was the last pandemic of a respiratory virus that compares to what we have now. I mean, of course, the HIV/AIDS emergence in the 1980s was a pandemic itself, and we've seen that has had, you know, staying power, but that we're also in a better place with that now. And we also saw, you know, when 1918 emerged, that the same serotype of influenza persisted until 1957, but it doesn't mean that the 1918 pandemic per se lasted until 57. So, you know, COVID is here to stay. We're not going to eradicate it, but we will mitigate it, and we will live with it. Mm-hmm. And in the long run, the variants that will emerge, net of their interaction with human populations, will cause less severe disease. And, you know, 50 years from now, I believe SARS-CoV-2 will be a cause of the common cold, the way that we have 4 coronaviruses today, which do cause the common cold. But how we get from today, where we're in the throes of what is still sort of the emergent phase of the pandemic, to that phase 50 years from now, where COVID is simply a nuisance virus, you know, is an area where we still don't know exactly how it's going to unfold. So stay tuned.
Good. Tom?
You know, I want to end on a positive note. I mean, we've had, you know, 12 months almost where it's just been, let's be honest, it's been miserable, right? But I mean, look how far we've come as not only a country, but as the world. We've developed vaccines in record time. This is going to be the precedent now, all right? We are vaccinating people. We're developing new ways to treat this virus. We're developing all these different models. Will there be bumps on the road? There will be, all right? But, you know, for everybody that's out there listening, there are so many people working on this. And, you know, what we've learned in 11 months on a novel virus is absolutely remarkable. And I think, you know, there's always silver linings. And I think the next pandemic, which there will be, you know, we will be poised. We'll be in a better spot and to rapidly respond. This kind of came out of nowhere. And as Andrew mentioned, you know, the great influenza, that was really something that was a long time ago. Who remembers that, right? Although there's some great books on this, but I'm optimistic, and we're going to get through this. So thank you very much, everyone, for coming to this evening's presentation.
Mike?
Well, I would echo everything that everyone said and say it's time now to let the science work, and let the science work in a positive way for us, because we've put it on the back burner for too long. I saw that Victoria joked about having Tony Fauci introduce us, but you know, that's the kind of recognition that we expect will help. If people hear this enough and they begin to see that it's working, that's what we need. And I think I do think it's beatable. I think it's taken longer than it should have, you know, to get really to get a handle on it. But you know, we've had other problems as well, and so you know, let's get beyond that.
Good. Ilhem?
Yeah, I think Mike stole my thunder. I was going to say, I think this has been as devastating as it has been, and as struggle, as much as we've all struggled. I really would like to I want to shine a light on science. Please believe in science. Talk to the experts. Listen to the experts. Be informed, and we have learned so much from this, and hopefully we will take away some really valuable lessons that will protect us in the next pandemic, because I know Tom said this came out of nowhere, but I would say that there's a lot of us who would disagree with that statement. And so I think this has been a really valuable lesson. I mean, the Democratic Republic of Congo is in its 11th outbreak of Ebola, all from new spillover events from the yet unknown reservoir Ebola virus into human population. So we are living zoonotic disease. This is the new reality, and we have to be prepared for it. So please go out there, support your scientists, make sure that their voices are heard, make sure that they have the tools that they need to go work on your behalf.
Well, well said. And you all stole all my thunder. So let me just conclude, first of all, by thanking all of you. We are deeply indebted to all of you, and in all by your research. We appreciate all the work that you have done on our behalf, so that this, that we do have a vaccine in pretty record time. I hope all of you out there that are part of the audience really had appreciated the opportunity to learn and witness firsthand the tremendous value that great research universities like UCI offer to the community. And you can see how these outstanding cutting edge researchers really rose to the occasion to solve some of the most pressing matters facing our society, and to do groundbreaking research. And we should just remind everyone that this is pretty dangerous stuff, right? Especially at the beginning stages. You all took a great risk in terms of undertaking this work on our behalf. So we are tremendously in your debt. This is why often many members of the community, hear me appeal to them for financial support. It's for reasons like COVID, so that we can pivot so quickly when an issue develops and confronts us as a society. And Matt, I don't know if you have time to put up the slide, but we are all very appreciative, and there's a lot of information about the pandemic. Our last year's Dean's report is available online. You could see it there. And if you're able to help and give the gift of discovery, please reach out to Andy or Robin, or contact us through the website. And you can see Andy and Robin, their pictures there, as well as their phone number and email addresses. And, we love doing these community forums. We think they're really what we're all about, is to share the great research that we develop here with you all. So please stay tuned for the next Dean's Distinguished Lecture in the spring. And on behalf of everyone, let me give the speakers and the panelists an outstanding round of applause. I appreciate all your wisdom and sharing your insight. We know how busy all of you are. So thank you, everyone, and stay well and healthy, and look forward to seeing you in person soon. Take care.